learning outcomes. After studying this module, you shall be able to know about genetic marker, its types and uses, the various techniques like RFLP, RAPD and AFLP, the various applications of these techniques. A genetic marker is an easily identifiable piece of genetic material, usually DNA, that can be used in the laboratory to tell a part of cells, individuals, populations or species. The use of genetic markers begins with extracting proteins or chemicals for biochemical markers or DNA for molecular markers from tissues of the plants, for example, seeds, foliage, pollen, sometimes woody tissues. Laboratory protocols, often well developed, but may need adjustments for certain species, are then applied, resulting typically in visual representations from staining or tagging techniques, which are then converted into data, usually allele types and frequencies or presence or absence of data. The various uses or applications of genetic markers are as: the genetic markers can be used to study the relationship between an inherited disease and its genetic cause. For example, a particular mutation of a gene that results in a defective protein. It is known that pieces of DNA that lie near each other on a chromosome tend to be inherited together. This property enables the use of a marker which can then be used to determine the precise inheritance pattern of the gene that has not yet been exactly localized. The genetic markers are employed in genealogical DNA testing for genetic genealogy to determine genetic distance between individuals or populations. Uniparental markers or mitochondrial or Y chromosomal DNA are studied for assessing maternal or paternal lineages. Autosomal markers are used for all ancestry. Genetic markers have to be easily identifiable, associated with a specific locus and highly polymorphic, because homozygous do not provide many information. Detection of the marker can be direct by RNA sequencing or indirect using alloenzymes. Some of the methods used to study the genome or phylogenetic are RFLP, amplified fragment length polymorphism, that is AFLP, RAPD, SSR. They can be used to create genetic maps or whatever organism is being studied. Now we will study the types of genetic markers. The modern genetics shows two types of genetic markers. First is the biochemical marker and second is the molecular marker. Now we will study about the biochemical marker. The biochemical marker detects the changes and variations at the gene level, such as amino acids, any hormone, enzyme, antibody or other substance that is detected in the urine or blood. The biochemical markers can further be classified as allozymes and isoenzymes. Now we will study about allozymes and isoenzymes. Allozymes, or also called as allozymes, are variant forms of an enzyme that are coded by different alleles at the same locus. These are opposed to isoenzymes, which are enzymes that perform the same function. but which are coded by genes located at different loci. They can be distinguished by electrophoresis. These enzymes generally perform very basic functions found commonly throughout all life forms, such as DNA polymerase, the enzyme that repairs and copies DNA. The significant changes in this enzyme reflect significant events in evolutionary history of organisms. The key to choosing which alloenzyme to use in a comparison between multiple species is to choose one that is as variable as possible while still being present in all the organisms. By comparing the amino acid sequence of the enzyme in the process, more amino acid similarities should be seen in species that are more closely related and fewer between those that are more distantly related. The less well conserved the enzyme is, the more amino acid differences will be present in even closely related species. The isoenzymes, also called as isozymes or more generally as multiple forms of enzymes, are enzymes that differ in amino acid sequences but catalyze the same chemical reaction. These enzymes usually display different kinetic parameters, example, different Km values, or different regulatory properties. The existence of isoenzymes permits the fine-tuning of metabolism to meet the particular needs of a given tissue or developmental stage. For example, lactate dehydrogenase or LDH. In biochemistry, isozymes or isoenzymes, also isoforms, closely related variants of the enzymes. In many cases, they are coded for by homologous genes that have diverged over time. Although, strictly speaking, the allozymes represent enzymes from different alleles of the same gene, and isozymes represent enzymes from different genes that process or catalyze the same reaction. The two words are usually used interchangeably. Now we will study about the molecular markers. A molecular marker is a molecule contained within a sample taken from an organism or the biological markers or other matters. It can be used to reveal certain characteristics about the respective source. DNA, for example, is a molecular marker containing information about the genetic disorders, genealogy, and the evolutionary history of life. Specific regions of the DNA or the genetic markers are used to diagnose the autosomal recessive genetic disorders. Molecular markers detects the changes or variations at the DNA level, such as the changes in nucleotide. The molecular markers consist of DNA markers, which is characterized as: first is the hybridization molecular based marker, and second is the PCR molecular based markers. First, we will study about the hybridization molecular based markers. The hybridization molecular based marker examines the differences in the size of the DNA fragments for which it requires high molecular weight DNA and probes and is usually performed on cellular genome. The two techniques which come under hybridization molecular based markers are the DNA-DNA hybridization and second is the RFLP technique. Now first, we will study about the DNA-DNA hybridization. In the molecular genetic technique of hybridization of DNA, single strands of DNA from two different species are allowed to join together to form hybrid double helixes. These hybrid segments of DNA can be used to determine the evolutionary relatedness of organisms by examining how similar or dissimilar the DNA base pair sequences are. The technique of DNA hybridization is based on two principles. The first, that double strands of DNA are held together by hydrogen bonds between the complementary base pairs, and the second is that the more closely related the two species are, the greater will be the number of complementary base pairs in the hybrid DNA. In other words, the degree of hybridization is proportional to the degree of similarity between the molecules of DNA from the two species. This process of hybridization is used in molecular biology laboratory to perform the techniques like Southern blotting and PCR. DNA hybridization technique is usually used to determine the genetic distance between the two species. Now we will study about the RFLP or the RFLP technique. RFLP, also known as RFLP or restriction fragment length polymorphism. The basic technique for detecting the RFLPs involves fragmenting a sample of DNA by a restriction enzyme, which can recognize and cut DNA wherever a specific short sequence occurs, and in a process known as restriction digest. The resulting DNA fragments are then separated by length through a process known as agarose gel electrophoresis and transferred to a membrane via the Southern blot procedure. Hybridization of the membrane to a labeled DNA probe then determines the length of the fragments which are complementary to the probe. An RFLP occurs when the length of a detected fragment varies between individuals. Each fragment length is considered an allele and can be used in genetic analysis. RFLP analysis may be subdivided into single, that is SLP, and multi-locus or MLP paradigms. Usually, the SLP method is preferred over the MLP method because it is more sensitive, easier to interpret, and capable of analyzing mixed DNA samples. Moreover, data can be generated even when the DNA is degraded. RFLP technique is expensive and a time-consuming method. The RFLP technique is a vital tool in determining the genetic disease, the gene mapping, and most important, to solve the paternity dispute cases. Next, we move on to the PCR-based markers. First off, let us learn what is PCR. The polymerase chain reaction, or the PCR, is a technology in molecular biology used to amplify a single copy or a few copies of pieces of DNA across several orders of magnitude, generating thousands to millions of copies of a particular DNA sequence. Developed in 1983 by Carey Millis, PCR is now a common and often indispensable technique used in medical and biological research labs for a variety of applications. Typically, PCR consists of a series of 20 to 40 repeated temperature changes called cycles, with each cycle commonly consisting of 2 to 3 discrete temperature steps, usually 3. The cycling is often preceded by a single temperature step at a high temperature, that is greater than 90 degrees centigrade, and followed by one hold at the end for final product extension or a brief storage. The temperatures used and the length of time they are applied in each cycle depend on a variety of parameters. These include the enzymes used for DNA synthesis, the concentration of divalent ions and the DNTPs in the reaction, and the melting temperatures of the primers. The first step is the initialization step. This step consists of heating the reaction to a temperature of 94 to 96 degrees centigrade, which is held for 1 to 9 minutes. Next is the denaturation step. This step is the first regular cycling event and consists of heating the reaction to 94 to 98 degrees Celsius for 20 to 30 seconds. It causes the DNA melting of the DNA template by disrupting the hydrogen bonds between the complementary bases, yielding single-stranded DNA molecules. Third is the annealing step. The reaction temperature is lowered to 50 to 65 degrees Celsius for 20 to 40 seconds, allowing annealing of the primers to the single-stranded DNA template. This temperature must be low enough to allow for hybridization of the primers to the strand, but high enough for the hybridization to be specific. That is, the primer should only bind to a perfectly complementary part of the template. If the temperature is too low, the primer could bind imperfectly. If it is too high, the primer may not bind. Typically, the annealing temperature is about 3 to 5 degrees Celsius. Below that, the team of the primers used. The stable DNA-DNA hydrogen bonds are only formed when the primer sequence very closely matches the template sequence. The polymerase binds to the primer template hybrid and begins the DNA formation. Next is the extension or the elongation step. The temperature at this step depends on the DNA polymerase used, that is the Taq polymerase, and it has its optimum activity temperature at 75 to 80 degrees Celsius, and commonly a temperature of 72 degrees centigrade is used with this enzyme. At this step, the DNA polymerase synthesizes a new DNA strand complementary to the DNA template strand by adding the DNTPs that are complementary to the template in 5' to 3' direction, condensing the 5' phosphate group of the DNTPs with the 3' hydroxyl group at the end of the nascent or the extending DNA strand. The extension time depends both on the DNA polymerase used and on the length of the DNA fragment to amplify. As a rule of thumb, its optimum temperature, the DNA polymerase polymerizes 1,000 base pairs per minute. Under optimum conditions, if there are no limitations due to limiting substrates or reagents at each extension step, the amount of DNA target is doubled, leading to exponential or geometric amplification of the specific DNA fragments. Next is the final elongation step. This single step is occasionally performed at a temperature of about 70 to 74 degrees centigrade, that is the temperature needed for optimal activity or for most of the polymerases used in the PCR reactions, for 5 to 15 minutes after the last PCR cycle, so as to ensure that any remaining single-stranded DNA is fully extended. And lastly, the final hold. This step at 4 to 15 degrees centigrade for an indefinite time may be employed for short-term storage of the reaction. The various PCR-based markers which will be discussed here are the SNPs, SSR, AFLP and RAPD. First is the SNP. SNP is a DNA sequence variation. Any two unrelated individual differs. It occurs when changes occurs in a nucleotide composition. Let us suppose that there are two DNA molecules with nucleotide composition. Both DNA molecule differs at a single nucleotide composition, that is C or G and A or T location, and that's why it is said as a single nucleotide polymorphism. Next, we will study about SSR or simple sequence repeats. SSR is a simple sequence repeat which is often termed as microsatellite, and STR, that is short tandem repeat. It is a sequence where 2 to 6 base pairs of nucleotide are repeated. These repeat units are generally di, tri, tetra, penta nucleotides. One common example of microsatellite where C and A are nucleotide which is repeated variable number of times. The variability of microsatellite is due to higher rate of mutation. Next, we move on to AFLP. AFLP is the most complex of marker technologies. It is a PCR-based tool used in genetics research and DNA fingerprinting. The procedure of AFLP technique is as follows. It involves the digestion of DNA with restriction enzymes, followed by ligation of adapters to the sticky end of the restriction fragments. The selected fragments are then amplified using two PCR primers. The amplified primers are then separated and visualized using the gel matrix. AFLP is used in molecular phylogenetic studies, creation of genetic maps, and AFLP is used in criminal and paternity tests to determine slight differences within populations. It is also used in the establishment of the linkage groups in crosses. Now we move on to RAPD or the random amplified polymorphic DNA. RAPD or random amplified polymorphic DNA is a type of PCR reaction by the segments of the DNA amplified in random. The DNA markers are developed by using random sequence of specific markers through the use of random primers. The RAPD is inexpensive, fast and easy method for detecting polymorphisms. The applications of RAPD are: it is used in genetic mapping. It is used to develop the genetic markers linked to a trait in question. It is also employed to study population and evolutionary genetics. Next, we see the class of the genetic marker characteristics in a table. The vertical column shows the characteristics, that is the number of loci, inheritance pattern, the positive features, the negative features, whereas the horizontal row shows the morphological markers, the protein markers, the RFLP markers, RAPD markers, and SSR markers. Next, we move on to the forensic significance. In forensic science, hybridization probes are used for the detection of short tandem repeats, and restriction fragment length polymorphism are used in DNA profiling of an individual to solve the paternity disputes. The genetic markers are also useful in gene mapping. It also helps in the characterization of the genetic diversity or breeding pattern in animals. Now, let us summarize all that we have studied in this module. The genetic marker can be said as any visible character or a gene or DNA sequence with a known location on chromosome. The various uses or applications of the genetic marker are as: firstly, it is used to estimate the relationships. Secondly, it plays an important role to find the diseases. Next, it is used to study the differences in the population. It is also used to describe the geographic patterns of genetic variation. Lastly, it is used to identify an individual or a species in a population. The modern genetics shows two types of genetic markers. The first is the biochemical marker and the second is the molecular marker. The biochemical marker detects the changes or variations at the gene level, such as the amino acids. The biochemical marker can be further classified into two classes, that is the allozymes and the isozymes. The molecular markers detects the changes or the variations at their DNA level, such as the changes in the nucleotide. The molecular marker consists of DNA marker, which is categorized as the hybridization based molecular marker and the PCR molecular based markers. The two techniques which come under hybridization molecular based markers are the DNA-DNA hybridization and the RFLP techniques. The various PCR based methods are SNP, SSR, AFLP and RAPT. Infrared science hybridization probes are used for detection of short tandem repeats, and restriction fragment length polymorphism are used in DNA profiling of an individual. It is also used to solve the paternity dispute cases. The genetic markers are also useful in gene mapping.